So, good morning, good evening, everyone. I'm pleased to welcome you today to our 5th webinar entitled The Co-Culture of Sensory Neurons and Keratinocytes. I'm Alexandre Guichard, PharmD PhD, specialized in clinical dermatology and product owner at NETRI. I'm in charge of the development of new models at NETRI. I was supposed to share the seat with my colleague Hélène Gauthier, neuroscientist and electrophysiologist, also product owner at NETRI, but unfortunately she is sick today, so I will do my best to reply to your questions. First, I will present you what we do at NETRI, then we will watch a testimonial from Stuart Prime, a group leader at Axel, our partner, and then we will have a Q&A session. NETRI, who we are. We believe in challenging the way we discover new treatments. We design, manufacture, and sell standardized neural organs on a chip. We intend to use neural activity at the network level to translate mode of action of any drugs. NETRI's vision of the future, the market is already looking to speed up preclinical phases, diminish clinical failure rate, and minimize animal testing. To address this, we believe the future of preclinical research will be about larger, faster, and more predictive data sets, and more importantly, generated from animal-free testing. Today, the models from academic are highly relevant, but low throughput. On the other hand, the standard P96 or P364 plates are high throughput compatible, but low relevant. Therefore, at NETRI, we are convinced that the future of organ on a chip will be about increasing relevance and complexity while maintaining high throughput capability by first democratizing organ on a chips, second, leveraging existing equipment, and third, standardizing data sets through digital library, and finally preserving and improving relevance. Here it's a presentation of our website on which you will find our technologies, products, services, applications, and capabilities. Today, I will focus my presentation on our capability to culture cell types and especially sensory neurons. I will focus my presentation today on skin innervation. Therefore, I would like to start with basics. The skin is a protective and innervated organ that decodes a wide range of tactile, thermal, and noxious stimuli. The perception of sensory stimuli involves various sensory receptors. The importance of these receptors has been underlined last year when the Nobel Prize in Physiology or Medicine has been attributed to Dr. Julius and Dr. Pataputian for their discoveries of skin receptors sensitive to chili pepper and hot, and touch, respectively. Here on the left side, you will see a macro representation of the skin, innervation, and on the right side, a micro representation, first on the skin, then spinal cord, and finally the brain. From an anatomical point of view, skin receptors are located into the skin, while neurons' cell body are located in the spinal cord. Then the signal is carried from the ganglion toward the appropriate integration center of the brain. In most of standard methods, cells are mixed together, which is not representative of the actual anatomy. Thanks to the compartmentalization, organ on a chip mimic the actual anatomy. They enable the co-culture and the connectivity between different cell types. For example, neuron-neuron, neuron-muscle, neuron-skin, skin endothelium with other architecture, and so on. Here it's an example of our first 2D model of innervated skin. The design of the chip is based on the anatomy. Our device uses channels to isolate cell population in compartments. In channel 1, here in green, we seed human primary keratinocytes. In channel 2, in purple here, we seed, channel 3, we seed a human iPS-derived sensory neurons from Axel. So we have neurons on purple channel, and between them, in the channel 2, the gray one, it's empty. It allows the increase of fluidic isolation. No cells in the middle. Between the channels, as you can see, we put micro channels, which allow the physical connectivity between the two compartments while avoiding any fluid leakage. Powered by the electrodes from Axion biosystem, we combine the power of compartimentalization with the power of electrophysiology. Here it is, our first chip, in which we co-culture skin and neurons. On the left side, human primary keratinocytes in green, and on the right side, the neurons derived from iPS from axol. As you can see, both cell types grew correctly in each compartment. If I zoom in the middle at the level of the microchannel, you clearly see that neurites in green start from the right compartments with the neurons, pass through the microchannels, and innervate the left compartment, which is also seen in transmitted light microscopy in the channel of keratinocytes. We clearly see the small black lines which correspond to the neurites. Here, another staining which reveals the TRPV1 receptors. The interest of our concept is double: to stimulate the skin, for example, with UV, chemicals, mediator release, stimulation like capsaicin, mechanical forces, and assess the outcome on neurons. But also, what is important is to do the reverse, to stimulate the neurons with mediators, with drugs, or with electricity, and assess what happens. There is a bug on the connection. Okay. The applications are broad. I'm frozen. Can you hear me? Can you see me? I'm frozen. Okay, so I'm frozen, but you can see me. The applications are broad. The pathophysiology, we can assess the pathophysiology of diseases, understand the mechanism of action and so on. But also with our chips, we can screen either the drug or cosmetic efficacy, but also the toxicity. And the field of application are also broad, especially in dermatology and cosmetology, for example, sensitive skin, pruritus, wound healing, in which keratinocytes and neurons are connected and involved, inflammatory dermatosis, such as psoriasis, eczema, or rosacea. The slides, just let me, yes. Our devices are compatible with standards and innovative readouts. Thanks to the transparency of our material, the PDMS, the microscopy is possible. Sorry, excuse me. Okay. One slide, sorry, I missed one slide before that. Yes. So I said our devices are compatible with standard, but also innovative device readouts. For example, since the PDMS is transparent, we can use microscopy, transmitted light, immunofluorescence, confocal, but also the supernatant can be sampled for dosage, for example, by ELISA or HPLC. Since the PDMS is soft, we can do histology, and we can also combine we can use our new technology like MEA to get new readout, innovative readout, the electrophysiology, which is the electrical activity of the neurons and which is correlated to a functional activity. If you want to know more about the possibility of MEA, I invite you to watch the video in replay of our 4th webinar presented by my colleague Serge Roux last week, an untitled functional activity. You will find the link on NETRI's LinkedIn page. So as explained as an introduction, at NETRI we are convinced that the high throughput is the future. Therefore, we provide our chip in our NeoBento format, which is SBS aligned on P96 well. Our format, NeoBento, is compatible with standard lab equipments. So I focused my presentation on skin innervation, but actually our models are versatile and agnostic of any cells. So we can apply, this model can be applied to neuropathic pain, nerve injury, and many other applications. And we can also replace the keratinocytes with other cell types, such as gut, dental or vaginal mucosa, Schwann cells, and so on. The technology allows this versatility. Here you have an example on this slide of one of our lines, the neurofluidic lines, in which we grow neurons with other cell types. But as you can see, the architecture, we developed 4 architecture for 9 products with different applications. If you want to know more about specifically organochip applied to sensitive skin, I invite you to read our article published last June in Cosmetics journal. Now it's time to listen to the testimonial of our partner, Stuart Prime from Axel. Just after, I will be back for a 10 minutes Q&A session.
Good afternoon, everybody. My name's Stuart Prime. I'm a team leader here at Axel Bioscience. Axel Bioscience Limited is committed to being a trusted supplier of human iPSC-derived cells, media products, and related contract services for life science discovery. Organ-on-a-chip devices are allowing industries to change the way they conduct product safety testing and discovery. NETRI has advanced the organ-on-a-chip microfluidic technology to a state-of-the-art level, which can be used in combination with an array of cell types. The Axel NETRI collaboration will provide the pharmaceutical and cosmetic industries with an innovative, high-throughput technology platform using NETRI's Dualink MEA microfluidic chip, combined with first-in-class cellular reagents from Axel Bioscience. For these industries, the selection of relevant and predictive in vitro models of the sensory response to skin to various stimuli is essential to assess products for the skin sensitization and toxicity market. In vitro models to date have typically used human cells with various animal neurons, such as rat, mouse, or pig. But to better represent human responses in innervated human skin models, the use of sensory neurons derived from human-induced pluripotent stem cells has enabled more relevant models to be developed. The current collaboration will therefore use Axel iPSC-derived sensory neurons, which express several voltage-gated sodium channels, such as NaV 1.7 and 1.8, associated with pain, as well as transient receptor potential ion channels, such as TRPV1, TRPA1, and TRPM8, that play key roles in nociception. In combination with human skin cells, the Axel NETRI collaboration will provide an in vitro platform suitable for investigating skin sensitization and chronic pain, as well as for identifying and testing drug targets and treatments for pain relief. Axel sensory neurons are utilized worldwide in both the pain and cosmetic markets, and through this collaboration with NETRI, we look forward to offering both this improved human relevant skin sensory model and future application of Axel's neuronal iPSC models across the NETRI platform. Thank you for listening.
Thank you, Stuart. So now it's time to open the Q&A session. So if you have any question, please ask them on the comment on the chat of this webinar. either I was very, very accurate and understandable, or I was completely not legible. So please ask at least one question. No questions? Okay, so no questions. So thank you for your time. So I just want to conclude with an introduction of our next and last webinar, the 22nd of last question just before the end. Are you planning on trying 3D culture in these organoid chips? This is a good question. We are developing a new architecture in which we indeed are we plan to co-culture 3D models. So either keratinocytes, different reconstructal epidermis, or we can also in this new chip add a biopsy or organoids, and this is something we are working on. And also BASF, one of our partner, has had a talk last month in a congress, and they presented the results of our chip with their cells, and typically they are creating a 3D culture. So yes, we are working on it, and soon we can release new information on this purpose. Another question: have you added immune cells to this model? Not yet on this model, but this is something we are working on also. Especially with endothelial cells. We are working on a chip with endothelial cells and a membrane and keratinocytes, for example. And in this chip, we plan to use human cells and assess the effect. But we didn't do on this particular chip human cells. Yes, we think, so the question is, can you induce dermatitis type models with cytokines? This is a plan. We didn't do yet because, again, the model I showed you, I presented you, is relatively new, but this is the next step. The first step was to build this model and validate the cell co-culture and the functionality. And the second step is to induce a disease. And of course, the first disease we will focus on is inflammatory diseases, such as dermatitis, for example, or with cytokines, with also like mediators, such as also with a stimulation like with capsaicin, for example. So yes, this is the next step, induce dermatitis. And the third step is to assess the efficacy of compounds to reduce this inflammation. Do you have any question? No. Okay, so I said just before, the 22nd of November, we will have our last webinar at NETRI based on neuropathic pain. So if you want to attend, it will be the same format. So we wait for you on the 22nd at 6 p.m. CET. Thanks for your time and for your attention, and see you soon. Bye.